And I also don't want to provide a detailed introduction to our today's speakers, because I know Irina quite well, but it's the first time me meeting Elan. But I know that Elan, you were a secretary of New York's Ecological Society, right?
Correct.
And you are also an expert in Myxomycetes in slime molds. So that's our speaker, Ellen, and then we will, I don't know if Irina or you who will be first today, but I will just introduce you, both of you, and Irina is a second, or like whatever will be the order of seminars today, but Irina is also an expert in slime molds, in Myxomycetes. I know your big part where Artzeria and Artzeriales group done with us in the FunDive project, but Irina is now based at the University of Tartu, right, in Estonia. And her research is dedicated mostly to the slime molds, but I know that not only for them. So I think what I will do now, if everything's work well, because Elan, you should have possibility to share your screen, right?
Yeah.
So maybe we can, yeah.
Hi,
What do you
Marta,
think, guys?
thank you.
Yeah.
We were discussing that. I think my presentation will be first, and Elan will continue in 20-ish minutes or so. So if that's okay.
Perfect.
Works for
Yes.
me.
Okay.
All right.
So
I'm
so
trying
yeah.
to
So
share my screen. You can
perfect.
hear me well, yes? Is that all right?
Yes.
Okay. Let's
Yes, we
try
hear
to
you,
share.
and yes. So I can leave the floor and everything to you. Thanks a lot, guys, for.
Could you please give me some indication of what you see on the screen?
Now it's loading, and now it's a full screen.
Full screen.
Through slime.
Okay, because I don't see myself, but okay, I will be talking to the
yeah, this
screen.
is, yeah, that's, yeah.
So should we start already? It's like 2 minutes past 9.
Yes.
Are we waiting or
I mean,
starting?
we can start. Because, yeah, I think we can start. But if you want to have some minutes to introduce yourself and like also to discuss something, then we can do it, guys. But I think we can start. I don't know if Glenn agrees as a co-host, but I guess we should start.
Right. Thank you. Thank you, Marta, for introduction. My name is Irina. I work currently at 2 universities in Estonia, in the University of Life Sciences and in the University of Tartu. And in both universities, I do study slime molds. In one place, I study the herbarium or fungarium collection, and in the other, I continue doing taxonomical work. But yes, let's talk more about our true slime. Although FunDive is more fungi, is more focused on studying of fungi. But today, let's take a step aside and dive into slime. And since slime molds are so cryptic and so secretive, I decided to make it a little bit like an investigation in this true slime style. So yes, bear with me. When people who have never met slime molds see them for the first time, they usually are confused and ask a very logical question, which is what the heck is that? And that's because slime molds usually appear out of nowhere, and then they can move, and they're also different colors, and they're so bright, so visible, and then they disappear as quickly as disappear into nowhere. And that is why in many languages and in many cultures, slime molds received common or vernacular names that reflect their secretive nature, like it's has something to do with witchcraft, usually with some forest creatures like wolf's milk or wolf's breasts or butter of some ghosts, witches, and even in Estonia, it's some species have a name as a name of tickspuu, which is crazy because it is quite large species. But of course, in reality, slime molds are not supernatural. Although they are very fascinating indeed. Their life cycle starts with spore, just like in fungi, but spore germinates with Myxamoeba or free-living amoeba that can also, if the conditions are, if the moisture is high, then they can become flagellates, and they can form microcysts. But the most interesting part is that if the resources are scarce, like the amoeba live in soil or in dead wood, but when they deplete the resources, they jump into temporarily, let's say, multicellularity. They can form multinucleate plasmodia that can actively move. And after some period of movement, the plasmodia form fruiting bodies that contain dry spores. And in this aspect, they resemble fungi. And that's why even now, slime mold research or Myxomycete research is the part of the general mycological research. That's why they are part of FunDive. So they eat bacteria, they eat algae, fungal spores, and so on, and they are microhabitats for for many organisms, such as beetles, slugs, nematodes. Usually the slime molds are studied, identified on the stage of a fruiting body. These fruiting bodies, plasmodia themselves have very little traits to note and to identify, but so the fruiting bodies are the most important part that we usually collect and study under the microscope and so on. There is a lot of different types of fruiting bodies, and they vary from millimeters to several dozens of centimeters even, so they could be quite large. Where to look for slime molds? The amoeba live in basically all terrestrial ecosystems, including sewage waters, but usually soil, dead wood, litter, and so on. And fruiting bodies, for fructification, they usually crawl up to some logs, dead logs, stumps, but also on they can fructify on living plants and in arid regions on cacti and so on. So that's usually how slime mold people look like. They move very, very slowly in the forest. They spend a lot of time on one place, and they usually bend over some dead logs and looking for very attentively looking for something. So that's how slime molds are found, just with a hand lens usually, and they are collected into suitable boxes. Match boxes are very nice for this purpose. And they are glued, the fruiting bodies are quite fragile, and they're usually glued to the bottom of the box. So the fruiting body, we are mostly interested in fruiting bodies. And but fruiting bodies can be tricky and deceitful in two principal ways. First of all, even though they are like better than plasmodia, they still have a limited number of morphological traits. You can see, for example, two species. One is very common Arceria stipata on the left, and on the right is a new species that is we are describing in the submitted paper. It's Arceria margaritacea, which are very, very similar and difficult to identify without applying much additional effort. And the second aspect is that fruiting bodies, as I showed, they are not formed by growth as fungi, but they are formed by transformation. from Plasmodia. And this transformation happens quite quickly, usually like sometimes in hours. It takes only hours. And so they're very heavily influenced by the environmental conditions that happen to be during these several hours. So they are, for example, their stalks can be very reduced or even absent, even though in normal fruiting body they should be present. Or caplicium can be abnormal, and so on. So fruiting bodies still can be very deceitful. So that is why barcoding is helpful, both in species identification, as I said, and also in species discovery. And I want to give two examples here. This is the example of Lycogala epidendrum, one of the most known species of slime molds in the world. And the genus Lycogala was studied by Dmytro Leontiev and his colleagues in Greifswald in recent years, and now over 40 new species are already described formally, and over 50 cryptic species are still undescribed. So that's the diversity revealed by molecular methods. And that's the usual, pretty typical picture when one morphospecies contains from 2 to several dozens of genetic clusters of sequences of genetic diversity, and these sequences correspond to either truly cryptic species that are undistinguishable, but also sometimes they are semi-cryptic, somewhat distinguishable, or even just overlooked species. Like you can see this Lycogala, they're very different. They can be identified. Some of them can be identified morphologically. But the other example is from my work, from this new manuscript submitted, but yet unpublished. I studied Arceria. You can see the type species, very common species, Arceria denudata, which, first of all, it does contain a lot of diversity, and I'm describing 6 new subspecies inside this species, but it also has a twin that occurs in neotropics, in Nearctics, and in Australia. Arceria denudatoides, that inside of this Arceria denudatoides, there are at least, we are formally describing 4 semi-cryptic species that can be distinguished, but with a lot of effort. You can see them with like scanning electron microscopy. So I mentioned geography here quite intentionally because earlier it was thought that slime molds are very cosmopolitic. So they form spores, spores can fly, so they should be everywhere. But that's more and more studies appear that show that it is not true. And we need to figure out where their ranges are limited, and we need to figure out where they live and why, and are there any endemic species, and so on. And for this purpose, and of course the ecology and niches, for this purpose, the limited resources that we have in slime mold research, this is not very well funded area, I would say, honestly. But luckily, we might ask for help from citizen science. And luckily, slime molds are not just cryptic and weird, but they are very beautiful, and they attract attention from citizen scientists and just from people. And there are several societies, or several informal groups, and also formal societies that are gathered around slime molds and are interested in slime mold research. For example, there is the biggest, I would say, community on Facebook, that this group, Slime Mold Identification and Appreciation, that has more than 80,000 participants now. And also there are several amazing photographers that attract, that have tens of thousands of followers, for example, like Sarah Lloyd from Tasmania or Alison Pollock from US. that is very recommended to visit and subscribe. This is just amazing. And also there is the country-wise groups that study slime molds and map and track slime molds. And also this, Ellen will talk about the New York Mycological Society. I will not stop here. But we can ask for help from all these people. And that's one example that I have from my work is the case of Spiromyxos lacanensis. It was when Tyson Ellers posted the photos of some unusual specimen on Facebook in this group, and we contacted, and he did microscopy and light microscopy, and we very quickly realized that it's something very unique. So he shared the specimen with me, and I sequenced it, and it appeared to be not just a new species, but a new genus, because it was intermediate between families Arceriaceae and Hemitrichiaceae, both in terms of morphology and genetically. So we described it as Spiromyxos lacanensis, which was named after its slocan belly, which, if I remember correctly, is like one of the favorite places of the collector. A very beautiful species. So I decided to try what will happen if we ask from citizen scientists to help. And within this FunDive project, I started citizen science campaign, which was called Read the Slime. And I focused on 3 target groups, which first was red arteria, then general Dianema and Dictydietalu. Sorry. I designed I posted the announcement on social media on FunDive website, and also designed, and there is the booklet, 12 or so pages booklet that you can see from FunDive website. Also there was a leaflets for printing and for sharing, and in FunDive we had some fun with activities such as FunDive bingo for student of the field mycology course, and just shared the information here and there. So the campaign was launched in the late July 2025, and here you can see the geography of obtained specimens. It was about 300 specimens. Of course, the FunDive is targeted to Europe, but when people wanted to send something outside of Europe, I didn't refuse. We discussed the terms, conditions, like collaboration and authorship and so on. And I got the specimens out of Europe also. So at this point, it's not finished. Of course, I databased less than 185 specimens. And at this point, I got barcoded 53 specimens. I mean, at least one sequence, and I try usually for more than one barcode. So for barcoding, I used FunDive protocols with slight modifications, such as, well, it was extraction with Chelex and aluminium oxide, PCR with group-specific primers, and then sequencing with nanopore, and sequence analysis. For making it more efficient, I used multiplexing. So I multiplexed. I pulled both different under one barcode. I pulled both taxonomically different groups, and for example, Arteria cinerea and Arteria denudata, like red and green, red and gray Arteria together, so I could disentangle them back. And also I pulled different markers for slime molds. We don't use ITS. It's not a very good marker for barcoding slime molds, but we use partial SSU, elongation factor, and the mitochondrial SSU genes. So one barcode usually had from, I would say, from 2 to 10 or 9 or 10 different products. Yeah. As for modifications, the important thing is when I process sequences, I removed ITS6 step from the pipeline, so because it's not ITS, it's not needed, and I matched sequences again against my custom sequence database. So I matched different genes, and I have results that both identifies the gene and the sequence. At the moment, Marta, Tisha, and I, we did 2 nanopore runs, and thank you, Marta, for that, for helping me in this sequencing. And I analyzed part of this sequences, 95 products, and in general, I have 119 sequences of different genes. And here is some previous results. Here you can see the main barcode for slime molds, SSU, partial SSU, which is quite a high success rate, I would say. And mitochondrial SSU also does pretty well, but like maybe a little bit worse. But elongation factor is the worst because it's single copy, it's quite long, and it's quite problematic. Not only this, but I had some challenges as well. Primers for slime molds not always work. There is this problem of introns in slime molds. They could be unexpected or some sequence variability. So not all specimens give nice PCR products. And we encountered this problem of bacterial contamination during the nanopore, so I had to cut out parts from the 5 and 3 ends, which corresponded to bacteria, and sometimes there are chimeric reads that also take time to disentangle and to fix. But most of the times I could get the needed sequence from it, but it just needs some time to process. This work is in progress, but about any discoveries made already, there is this cases. There is the species of Heterotrichia insignis, former Ratzeria insignis, that was thought to be cosmopolitan, but I figured out that in fact it is 2 species, and we don't have Heterotrichia insignis in Europe, but the true insignis actually occurs in Paleo and Neotropics, and in Europe we have sister species, other Heterotrichia, which I will name as Heterotrichia nedilco, which is slightly larger than insignis and form larger colonies, but is still similar to it. And during FunDive campaign, I got a lot of specimens of nedilco from Europe, and they are all not true insignis. So I assume that it doesn't occur in Europe or occurs quite rarely. So okay, thank you. Now my part is done, and I give floor to Ellen.
That was awesome. Thank you, Irina. So much to learn from you. It's unreal. Let me go ahead and share my screen. All right. Do you guys see what you need to see?
Yes, we see. Perfect. Thanks.
All right. So thank you, Marta, for the introduction earlier on. I appreciate the fact that you called me an expert, but seeing the list of attendees, there are definitely a few people in the audience who are a few notches above me and definitely a few publications ahead of me, since I don't have any currently. So my talk is going to be talking about the invisible, the invisible majority. Most biodiversity is small and overlooked and everywhere. So some of the most extraordinary organisms in New York City are actually smaller than a grain of rice. Fungi are actually closer to animals than plants, and slime molds aren't even fungi. But New York City is definitely a biodiversity of a hotspot. So my talk is going to be about the biodiversity, as well as citizen scientists and what my club specifically does on the walks that we go on. So what even is a slime mold? They are these weird, enigmatic organisms that kind of creep along the forest floors, changing their structures and their colors into these beautiful, magnificently teeny structures. But that's the reason why many of us love them. So I wanted to share with you guys a quick time lapse of one of these slime molds that I found in New York City parks. I brought it home. Here we can see it changing its colors and the cytoplasmic streaming, having the structure being pushed upwards, and you'll see it start to kind of morph and trim down as it begins to eventually change colors from this white color that you'll see when it's immature. And as it matures, it begins to become darker and darker. But unfortunately, because I have a cat who decided to play around with my time lapse, that's all you get. One other time lapse I can show you real quick to kind of give you guys an understanding are this species of Stemonitis. They oftentimes appear in an array of colors when they're immature. They can be yellow, they can be white, some are even pink, but that doesn't necessarily give us an indicator of the species itself. There's a whole lot of other things that you got to do, from measurements of the entire sporotheca to the stipe, to the stalk, even the spore size, the ornamentation, the nets. There's a whole lot of work that goes into identifying most Myxomycetes that require microscopy. So just wanted to jump through these two slides real quick, jump to this one, and talk about this, because at first glance, these two organisms can look remarkably similar. On the left, we have an earth star, which was found growing on Fire Island in New York in 2023 in a mulch pile along a sidewalk. On the right, we have Diderma asteroides, which were found growing on the back of a leaf, and they're roughly 1/100th the size of the earth star that we just saw. They're not fungal, but they are a eukaryotic organism in the kingdom Protozoan. This is a Myxomycete, or colloquially known by its common name, slime mold. Both form structures that split open into these star-like rays, exposing a central spore mass. The resemblance is striking enough that people often assume that these two different kingdoms are closely related. There's a lot of similarities between Myxomycetes and fungi, which is why they used to be classified in the fungi kingdom. Wanted to talk a little bit about the structures of a myxo. Here we can see the sporotheca, which is the spore-bearing part of the fruiting body that contains the spores and the capolitium. They come in many shapes, sizes, and colors. The sporangium or the sporocarp is the entire fruiting body. The peridium has many colors, textures, patterns that may be persistent or evanescent. And then the hypothalamus is a very thin membrane or layer of material produced during the plasmodial stage that acts as an anchor or glue for the sporocarp to adhere to the substrate. And then the capolitium are these fine threads that are filamentous or hair-like. They can also be calcified. They also often contain ornamentation such as spiral bands, spines, cogs, or even rings. The stalk and stipe is the supporting structure of the sporangium that elevates the sporotheca above the substrate to aid in the spore dispersal. And then here we see also what I like to call a pseudo-columella or a false columella. It looks like a central columella and acts like one, but it isn't actually really one. It's generally made up of a different type of matter from the spore mass. So I went to the Myxo Congress back in 2023. And before that, I had no training whatsoever or background in slime molds. I was, like many of you, just very curious. I had only ever uploaded 2 species to iNaturalist, and honestly, I wasn't even sure what I was looking at yet. This map shows all the Myxomycete observations in New York City before 2020 that were mine. Just 2 lonely pins. That's all I had. It was a start, but I had no idea how much I was about to uncover, and I wasn't the only one in the dark. Here's a map showing all the slime mold observations in New York City before 2020 from all users. Even with amateur naturalists, scientists, and a city full of mushroom lovers, there's still very few observations and people looking for, hunting for, and identifying Myxomycetes. Slime molds just weren't being noticed. And there were people who were really good at looking for them. I just don't think they knew what they were looking for. And a lot of these Myxomycetes were just hiding in plain sight. Since then, overall, we found nearly 90 species with 2.7 thousand observations and over 527 observers. To date, I personally have found 71 species in New York City proper alone. Overall, we have found over nearly 90 species, which is 3 times the amount since 2020. And the observations and number of observers have multiplied 5 times. So why do we, the New York Mycological Society, push so heavily to use the iNaturalist platform? I like to view it as kind of like a sticker book for adults. It's great. It crowdsources. It's free and it's high quality. And there's Android and iPhone apps. You can also crowdsource the identifications and solicit feedback from experts who are some of them who are in the talks here. You can also add observation fields and notes to your specimens to help you aid in retrieval of them. And you can also join projects and that kind of things. One of the projects that I started was called the Slime Moles of New York City. You can contribute to them and kind of see if you're a leader in these projects. In addition, the really cool thing about iNaturalist is every year it gives you a year in review. It doesn't just catalog the Myxomycetes, it catalogs everything you've done, how many observations you made, how many identifications you've made, the different kingdoms, all the way down to the granular level of species. It's really cool data descriptions that show you all the stuff that you need to see. So how do we do these walks that we're talking about? Well, my club, we post all of our walks that we call pop-ups the week of. Other walks may be labeled as a non-New York mycological event, which we denote, but we also promote them. Lastly, we do have scheduled walks that take place in the late spring into the late fall. These are usually announced and posted well in advance. So here's an example of the event page itself where we promote the location, along with Google Maps, directions, who the walk leader is, and their contact information for those who get lost or might be arriving late. We also provide an up-to-date synopsis of what people are going to or should expect on the walks. Given the locale, we provide weather forecasts as well as recent rainfall and the time of year. Lastly, we also provide written instructions for those coming by public transit, along with a description of where exactly we plan to meet. And it should be noted that we also pride ourselves in making sure that all of our walks are easily accessible by public transit. And of course, we also provide detailed instructions on how to use iNaturalist, which the club relies heavily for documentation. Before any of our walks start, we usually wait 10 to 15 minutes for those coming by public transit, as there's oftentimes delays. But we also make sure that we take a group photo before the walk begins. This helps us with identifying a bias with recorded species. For example, when Myxomycetes peak, it's usually because I'm on the walk. After the walk ends, we like to promote and knowledge share and cross-collaborate with our club recorder, who tries to attend nearly all of our walks. What the recorder does is during the walks, we go up to him and we'll let him know what we found. He'll then email us a comprehensive list of all the species that we found during the walks. And then we, the leadership of the club, we'd like to invite all the members to be part of that email to help foster knowledge sharing and cross-collaboration, to help other people begin to learn the species names of all the different fungi and Myxomycetes we find on our walks. So what do we do with this data? Well, we take all the data that we have from all the walks and we put it into a database that's pretty comprehensive. It exists on walklists.newyorkmyc.org. And you can see here, you can search by walks, you can sort by park, and there's even species lists. Recently, the club recorder took it a bit further by breaking it down and providing tools to actually dive deeper into the data. This allows anybody, which is open to the public, to peruse our data to kind of create and generate reports. It's like everything from the time of year, and you can bookend it to say you only want to see a specific year or a specific date, to months, to species, to larger genre. You can see that you can even filter by the observations to see what you find most often. You can even sort by park, and you can even create these cool little graphs that will show you all the data. So here's an example real quick to kind of show you some of the data. We can kind of see where the Myxomycetes peaked. This is around the time when I kind of joined and really started looking at Myxomycetes in the club. And one other thing that I know Irina spoke about it, matchboxes. Alison Pollock turned me on to these paper boxes as well. I decided to create this little thing I call a slime vault that you can store all the boxes in for easy retrieval and storage. And that's pretty much all I have for you guys. You can also contact and follow me. I did decide to make a little AI action figure might be a little bit dated now, but I thought it'd be fun to share with you guys. And I appreciate you guys inviting me to give this talk today.
Thank you very much. Thanks a lot for your talk. I still can't believe that. I mean, have never seen this slime mold that is like very similar with the shape to Geastrum, to the Geastrum species, and I'm like, oh my God, I could not believe that it's even possible that. But it looks like very similar things, so people can really confused. I think I can start the discussion because I saw one question in the chat. So either Mada Sibero will ask or I can maybe read aloud the questions. I think the question is more to Irina, but maybe it's for both of you because it's on the new speech, or maybe you can probably see the question in the chat as well, right? So maybe it will be easier then. But if someone cannot see it, it's like the first question out of 3 is what is the minimum percentage of the similarity required to like with the reference strain for DNA barcoding that will indicate a new species? So I think it's a tricky question. So I don't know, Irina, probably you are now working with such a thing, so you can answer the question.
Yeah,
I think.
thank you, Marta. Yes, indeed, it is a tricky question, and there is no universal similarity or dissimilarity threshold for it to be definitely a new species. There are rather local barcoding gaps between the clusters of sequences, and sometimes in some groups species are quite recent, so quite young, so to say, and so they're quite close, but still there is the gap, and they're divergent in morphologically and also genetically, but this gap is quite narrow. But for in other groups, the whole genetic structure is so different. Like, for example, in dark spore slime molds, usually these distances are smaller. For bright spore slime molds, these distances are quite high between different genetic clusters. So there is no universal number, and even sometimes there is the thing of no clear barcoding gap between species at all. So there are some ribotypes or some genotypes that are somewhere like alleles that migrated that conserved in some populations or in some species, and so they kind of spoil the picture of like clear separation between genetic clusters. So it's a difficult question of species delimitation, not only in slime molds, but in fungi also, and in animals as well. But there were other 2 questions. Is there any specific biochemical or physiological test to determine novel species, or is it based on genome and morphology? As far as I know, there is no biochemical tests, but there are the slime molds can be cultured. They are cultivable. At least some species of them are. So there could be done this tests like mating or crossing, like mating tests, where they check if this 2 plasmodia can fuse and then form fruiting bodies, or they do not fuse. So that's kind of the checking of the biological species concept here, but it's also very time consuming and resource. It's not feasible to do for every species of slime molds. It's like a lot of work. But yeah, morphology and phylogeny now are the most crucial, or also now the ecology and biogeography. Sometimes these lines of evidence are helpful. But yes, these two are principal. And also, do we need to compare the candidate novel species to the type strain at the same growth condition, like if we want to describe novel bacterial and fungal species? Usually, at least nowadays, new species are described without cultivation. They can be cultured, but they are not. So we don't compare with the strain. But of course, as Slimal taxonomy follows the botanical code of nomenclature, so we have type specimens that are kept in herbaria, usually in dry conditions, and we compare novel species to type specimens if they are preserved, which is not always the true. Sometimes they are lost, many times or they are very, very old, and there is nothing to compare with. But yes, we should compare with type specimens.
So I can also like add something on this bacterial, how to describe the novel bacterial strain, or for example, things that are bacterial and uncultured. There are many, many different endosymbionts of different invertebrates like insects, and they cannot be cultured on the plates or in the cultures. And we also describe like new, let's say, species or monophyletic lines out of it. And then there are some different resolution, like I would say, ways to describe it, and some are still in the form of like name of the species and candidatus, and for some it's just like unculturable organism that is just called by the new name or new strain or line name. Yeah, so if there is any more question, you can raise your hand or just turn on your microphone and feel free to ask. Thanks also, Ellen, for sharing this plastic, like 3D print plastic thing that I remember Irina showed me her collection, and also this matchboxes thing in the lab, and that was really nice way of keeping the specimens in a tidy or organized way. So it's a good thing to use in the lab. Irina, there's a question for you, whether you still accept specimens.
Thank you for the question. I promised to myself that I'm not anymore. But if it's something very, very interesting, then
so
maybe.
Yes, the answer is yes.
It depends on what are we talking about. Not red arteria probably, but Dictydietalium and DNM especially. I would accept. Yes, I do accept.
Okay. I can have, if I can let myself ask a question, I have a question to Ellen, because it looks like there's like a lot of great work you are doing as a community, and you've mentioned that the reports are kind of used as the official reports as well for the species diversity, or because it looks like you're documenting the area very well, and there's like the updates quite regularly by many people involved. And is this like really providing like the biodiversity monitoring sort of outcome, yeah.
Yeah, great question, and thank you for it, Marta. Yes, we do generate official reports. Part of the agreement that we have with New York City Parks here in New York City is to provide them a biodiversity report yearly. Our former club president, who I believe is also in the audience today, Sigrid Jacob, has been instrumental in putting those reports together on behalf of the club, and she continues to do so today. There's a lot of input that comes from the club. Yearly, we also do something called a taxonomic star chamber, where we get together with the quote unquote leaders of the field in our group, and we discuss what new taxonomic names we want to refer to and which ones have been moved around and changed around. So we kind of go through and we come to a consensus on what is currently the standard for Myxomycete names.
Oh, that's really nice. That's really cool. It sounds like very motivated organization and like motivated people to really do the. But that's really nice. I see, Irina, there's more questions to the DNA in the chat. So yeah, I don't know if I can like tell you or you can also read them, then it will be probably, yeah. So
first of all, I wanted to add that I also printed this slime vaults. So we started printing them at the University of Tartu, currently in small scale, but we already figured out how to print and modified it a little bit to match our matchboxes because they're a little bit wider, but we managed to do it. Thank you. It's a really cool way to store the specimens.
Are there differences between the matchbox size?
Yes,
that's crazy. I didn't know that. I mean, I thought that maybe it's a narrow thinking.
Different
Sorry.
Much. But quickly to answer the questions, how well DNA is preserved in old specimens? My absolute record was about like I think 45 years, but it's very rare case. Usually about 20 years. If it's more than 20 years, it makes it really difficult. Not impossible, but much more difficult. Okay. Do you think it would be beneficial to systematically look at the bacteria communities with slime mold specimen DNA extracts? This is a very good question. Thank you. Yes, it's a very interesting idea. I think there is a lot of, I know that there is a lot of other DNA except slime molds, and I never specifically looked at anything else than slime molds in these extracts. But sometimes, occasionally, like by mistake, there is other sequences appear, but I usually discard them, but I never try to analyze them. But it's very interesting idea. Then there is a question about what is the current situation with phylogenomic and functional genomic studies on Myxomycete. Phylogenomic is developing very rapidly. There are studies, several new studies on dark sport slime molds on this clade that involved several hundred genes. So, and I know that new studies are emerging, so I hope that they will be published soon, and this is emerging. But functional genomics, not that I know of, but I might be just not following this direction. So I don't know about the functional genomics, but it's of course something that would be interesting, very interesting to do.
For slime molds, I don't recall any like comprehensive studies on functional genomics, but I guess that there are some research, but it's, yeah, I haven't seen any big paper that is like out in some time. So I see, Irina, there are more questions to you, but also to Ellen.
Cytochrome oxidase, the question about cytochrome oxidase, as for me, my experience, in my experience, somehow it's very tricky gene to sequence. I tried many combinations of primers, designed primers for Syria, and it doesn't work. So I just refused to spend more time. So instead I took mitochondrial SSU as additional marker. So I have 3, and it's quite for most of the purposes, it's good enough. But then if we need to sequence more, then I would just think I moved to phylogenomics level instead of sequencing manually more and more barcodes. And there is also some comment about the US, so maybe it's to Ellen.
Yeah, it's like about the matchboxes that comes with different sizes and level of durability, I would say. But I have one more question to Ellen. Is that because it's like slime molds are also like in the US, right? Considered like they are included within the mycology in general, right? And the mycological societies, they are also associating slime mold people. And I mean, I know that slime molds phylogenetically are quite distant from fungi in general. So, but it's like let's say they share kind of lifestyle with fungi, and it's like some things that are kind of bringing them up together, the field work that is needed for both, let's say, groups is quite comparable. But I'm wondering whether, because in Europe I know we have very few slime mold communities that are really slime mold specific, that will include, let's say, citizen scientists or non-academic experts. But I'm wondering how it is in the North America. Is this also like part of?
Yeah, it's a good question. I know a lot of people in mycological societies kind of look at slime molds. A lot of people also kind of refer to them as like the stepchild of the societies. But they are very intrinsic in our culture and our societies. I have personally seen an uptick in the documentation and the interests people are having with finding and documenting slime molds. I think a lot of that can probably be attributed to the photography on Instagram with the people that Irina mentioned, as well as the Facebook group, and also the openness of the societies and the Facebook slime mold groups to accept people and help teach people is also kind of boosting the interest here in the US.
Okay, it's like, for me, it's very, yeah, it's like I don't know that many slime mold people, so I'm very glad to hear some stories from this world. Yeah.
I'm also hopeful that this year the Congress, which last time happened 3 years ago in Estonia, it's happening in my backyard in New Jersey. So this could be a stone throws away for me to take the train across the bridge into Jersey to attend it. I'm hopeful that some of you here in the audience will be attending this year. I'm going to do my best to attend it, but I do have some personal stuff that's going on that might limit my availability. But I'm hopeful to get out there at least a few days this Congress.
Cool. So yeah, maybe we can provide extra announcement attached to this webinar recording that there is like a upcoming meeting for slime mold, so people can be more aware that it's coming. Yeah, Irina, you will be there, or you will have other plans? I don't know if Irina will join, but for sure, let's hope. Oh, great. Okay, so I don't know if there are more questions. I see the discussion about the universal, so-called universal primers for metabarcoding, and I see that no, and that there are some more complicated things related to that. So I don't know if there's any more question somewhere, or is there hidden question or someone is raising hand? I don't see anything. So I don't know if we want to slowly stop the meeting and we can close our webinar, or I don't know if there's anyone who wants to ask a question. That would be the last moment, and we can officially stop the recording and we can have less serious discussions. No, obviously joking, but I don't know if Glenn will agree that we are finished now.
I think I'll make a small recap, and then if by the end of that short recap nobody has asked any question, then I guess we can call it a night. Yeah. So thank you both to our speakers. I could not join in time to do some of the technical go through some of the technical stuff with you because I had some technical problems on my own. But this was really nice. I don't know too much about slime molds, I guess. So it's good to at least get the freshener again, see what the life cycle is, what all of the different fruiting bodies types are. And it is really nice that also Elin showed that this type of convergent evolution exists between the earth stars, but also the slime molds. And it made me also think of the Rafflesia flowers. Those parasitic flowers that kind of look the same as these hiastrums, and now I know also that they look a bit like their slime molds. So really cool that there's also a lot of new species still to be discovered, and even if we think that we know some of the most common species in Europe, like these Assyrias, and it turns out that it's not really a European thing. It makes me think of Clavaria solingeri, which we all thought we knew, and but it's apparently some species from Java. I mean, there's so many parallels to draw between fungi and slime molds. So it makes sense to me that it's kind of the same people looking at them. So yeah, really cool. What you are doing, Elon, in New York is amazing. I guess in only 4 years' time, you almost increased tenfold the number of observations, and most of them are, of course, by you. But I'm guessing that you also inspire a lot of people to do the same. So this will snowball for sure. In a few years, you'll have 20,000, 30,000, 50,000 observations, and New York will be the best observed area on Earth for slime molds. So that's quite nice to see. Nice that you also record everything. And I guess this might be a question to you. If everything ends up on GBIF, do you put everything on iNaturalist? Is this somehow like up on the internet? Well, except for your or next to your general species lists.
I'm sorry. What's the question?
If you record all of your species, right? And so
Yep.
do you? So I saw the lists that you all made, or that you sent through to each other by email. So do these also end up then back into iNaturalist so that they somehow all end up into GBIF?
Yes, it's a great question. So I would say about 80 to maybe 90% of the observations in that list make it to iNaturalist. Oftentimes, and maybe that number is being conservative, it's not necessarily every single person documenting it to iNaturalist. So if we see like the super common ones, for example, like we oftentimes don't make another observation of the ones that we see day to day on iNat. But the ones that are really interesting and cool looking, yes, we do make observations to iNaturalist. And in our walk lists, when we reply back to the recorder and we include the link to the iNaturalist observation, we include that in our database. So you can actually see on the day the actual observation on iNaturalist.
Yeah, cool. Thanks. That's really nice that you make the effort to also document it back to iNaturalist, at least for most observations. And then what I also thought was kind of amazing is the slime vault that you created. I guess we'll see in the future many more of these 3D printed gadgets for mycology and for or mycologists in general. So it's nice to see an example of that. So with that, I guess there are no more real questions. For people who are trying to reach out to Irina or to Elan, you can find their details on the registration page or on FunDive, or I guess, you both are quite easy to find. So do please
also,
email them.
Yes, also Elan shared the link on the chat, so you can find it. Sorry.
Okay, thank you, Marta and Glenn, for organizing the seminars, FunDive seminars. It was really nice to see you all, and thank you all for listening and coming to hear about slime molds. I hope you love them now as much as we do.
Yeah, I see that there's a big discussion on which primers, like which markers to just. So it's like I see that there is like a big and also the big potential in this field, and also like a lot of unknowns, and also you have some. It's Mada, right, from New Zealand, that maybe will start doing research on slime mold somewhere there. So it's like really cool that the message is spread. And I will have to check this functional genomics. I really kind of overlooked that. I try to follow some sort of phylogenomic research on different organisms like that, but I haven't noticed anything. It's like barely anything on Ascomycetes in past years, so I'm not sure whether. Yeah, but it's really great, and I'm very thankful for your talks and for the discussion. And I guess we will all kind of stay in touch with asking each other or discussing different issues and stuff. Yeah.
Indeed. Thanks, everybody.
Thank
Okay.
you. So I think we
Thank
can
you, guys. Have a
officially
great
close
evening.
the. Yeah, thanks and have a
Bye.
nice day or evening. And thanks again for your work and presentations.
Bye.